speaker is Chantal Vogels from Yale, who's going to talk about impact of dengue virus genetic diversity on inhibition of Wolbachia.
Thank you, and thank you to the organizers for the opportunity to present our work here today. So when asked the question of like, are all dengue virus strain serotypes equal? I'm going to agree with Anh Yong that I don't think they are. And really the perspective that I'm going to take is the perspective of control interventions. And specifically, I'll be talking about impact of dengue virus genetic diversity on inhibition by Wolbachia. So we heard a bit about Wolbachia yesterday already, so I kind of just want to touch upon sort of bigger concepts of how Wolbachia-based control of dengue works. Wolbachia is basically used in two sort of distinct strategies. First strategy is you release male mosquitoes in the field that carry Wolbachia, and this basically leads to population suppression. And that is because if the Wolbachia-carrying males mate with wild-type females, they cannot get offspring due to cytoplasmic incompatibility. And so this is really a way to kind of like dampen your population, but you're not necessarily establishing Wolbachia in your local population. So the moment you basically stop releasing the males, you have the chance that the population would bounce back. The second strategy is population replacement. And key here is that you release the female mosquitoes that carry Wolbachia, because those can have offspring with both Wolbachia-carrying and wild-type male mosquitoes, and through maternal transmission basically helps to get introgression of Wolbachia in your local population. And with presence of Wolbachia, what a large body of studies have shown is that virus transmission is generally inhibited. We've heard some exceptions yesterday. The perspective that we wanted to take here is that a lot of the dengue studies that have been done in the lab, sort of evaluating effectiveness of Wolbachia, is that they typically take like 1 or 2 strains per serotype and then conclude if there is strong inhibition, okay, it works for this serotype. But as we've now heard a couple times, dengue is really, very genetically diverse. We have the 4 serotypes, but even within, we have several genotypes and major minor lineages. So there's a lot of genetic diversity within, and so the question we had, like, how is that going to impact Wolbachia-based control? So in these trees over here, you see red circles. Those were viruses that we selected for experimental evaluation. And so we really tried to have like a very diverse panel. We ended up with 60 viruses, so 20 dengue 1s, 20 dengue 2s, 10 dengue 3s, and 10 dengue 4s. These came all from the Yale Arbovirus Research Unit and the World Reference Center. And with that, we basically set up a experimental system so we could really sort of comprehensively evaluate how well all these different viruses would be inhibited by Wolbachia. This is the work led by a PhD student in my group, Afish Odende. So he took on this major challenge, basically doing vector competence experiments with 60 viruses. And it's not ending there, because we also wanted to include different Wolbachia strains. So we have in our system a wild-type mosquito. So this is Aedes aegypti from Australia. Then our collaborator, Perrin Ross, basically created these 2 different Wolbachia strains. So one is a WMEL. It's a specific strain of WMEL, so that's why you will see it's called WMEL-M. We heard some discussion around like heat stability and tolerance yesterday. So this particular strain of WML has like a slightly better heat tolerance than some of the other WMLs. And then we also wanted to include a WLB, which is also kind of known for sort of higher heat resistance, which could be useful in the field, especially in warmer areas. We then infected those 3 colonies with every virus, so we did it in parallel, so they were all exposed to the same virus. Challenging thing with dengue and mosquito studies is that if you use fresh virus, you get best infection rates, so that's what we did. Which makes it a lot harder to do these experiments because we cannot necessarily control titers all that well. So we tested everything in parallel, and then after 10 days of incubation, we tested mosquito bodies for infection, legs for dissemination. And just to kind of make the point of like why we selected those tissues. So here you see kind of like a cross-section of a mosquito. Mosquito takes a blood meal, virus ends up in the midgut. That mosquito midgut is really like the main barrier that the virus needs to overcome before it can sort of start spreading throughout the mosquito body, infect salivary glands, and then ultimately can be transmitted. So in our case, when we tested basically the entire body, it's an indication that at the minimum, the virus was able to get into the midgut. So that's our measure of infection. And we use legs as a proxy for dissemination as a proxy for transmission. And that is mostly because some of our collaborators found that that's a better proxy for transmission than trying to collect saliva. And so that's basically the two main rates that we determined in this study. I'm going to start with showing data for infection. It's probably a little bit hard to read, but basically every row is a different virus, so it's the 60 viruses. And then on the x-axis, you see infection rate. The first data that I'm showing is for wild-type mosquitoes. So even without Wolbachia present, we actually see a huge variation in how well this particular mosquito population is infected by this diverse set of strains. And so that's really a key point to realize that even sort of in natural transmission, there can be a huge difference in like how well a particular population can transmit genetically diverse viruses. That was, of course, not our main factor of interest. So if we now look at the Wolbachia results, so the middle column is WLB, the column on the right is WML, and then from top to bottom, we have dengue 1 in blue, dengue 2 in purple, dengue 3 in orange, and dengue 4 in brown. And you can already start to see that like there's definitely some level of inhibition as compared to wild type. If I then also pull up the dissemination results, which is sort of our closest proxy to transmission and the most epidemiologically relevant data, we see overall that there's, again, variation, both between the two Wolbachia strains, as well as between the different dengue serotypes. And so I guess one thing that pretty much stands out is if you look at dengue 2 in purple, we see very low dissemination rates in WML, basically meaning that WML is very effectively blocking dengue 2, at least the strains that we tested. And on the contrary, dengue 3 stands out because we see the highest dissemination, not just in WML, but also in WLB. But given that we're already starting with variation in wild type, we kind of wanted to account for that to make a slightly more fair comparison, because you can imagine if you're trying to compare viruses that are very differently susceptible in the wild type, that's sort of like a hard comparison. So what we did for the next set of graphs is we basically determined the relative dissemination, and we're splitting the data up in different ways. But basically what this means is that we do relative dissemination as compared to wild type. Meaning that if your relative dissemination is 1, then in presence of either of the Wolbachia strains, your dissemination is basically equal, so there's no inhibition. If the value is 0, it basically means complete blocking as compared to wild type. And so if we first sort of split up the data by the two Wolbachia types, we see sort of a more intermediate inhibition by WLB and overall a stronger inhibition by WML. But again, there is still a huge range. And so to take this one level deeper, I'm basically showing the same data. So again, every dot is like one virus that we tested. Now we're breaking it down by the two Wolbachia types as well as the 4 serotypes within. And now again, we're kind of starting to see some patterns. I think the main takeaway is that for any of the serotypes, there is a lot of variation. But particularly in dengue 3, we see the most strains with sort of like the least inhibition, which could be concerning. We took it one level further to go to the genotype level. So here you're really starting to see again, like if you really break it down by all the different genotypes within the serotypes. We obviously did not have as much data points here because for some we had only like 1 or 2 virus strains that belonged to a particular genotype. Others we had more. But I think this again sort of brings the point across. So even if we look at more granular levels, so even sub-serotype, there is that variation. And so it's actually really hard to draw conclusions for either an entire serotype or an entire genotype, given that sort of between strain variation that we're seeing. You can imagine if we had picked, let's say we take genotype 3 within serotype 3 as an example, if we had picked one of those upper dots, only did our experiments with that virus, we probably would have concluded, oh, there's not much inhibition, whereas if we picked one of the lower ones, we would have made a completely different conclusion. So I think this really drives the point home of like selection of whatever strain you use in your experiments matters. We then also looked at some other factors. Like I mentioned, we used fresh virus in these experiments, and as a result, we were not able to control for the virus titer that went into the blood meal up front. And so what you're seeing here on the x-axis is basically how much virus we added in the blood meal. And then on the y-axis is, again, that dissemination rate. If we look at the wild-type mosquitoes, is in that teal color, we see a pretty clear correlation that if you give more virus in the blood meal to these mosquitoes, you basically just have a higher chance that they become infected and disseminated. This is something we know pretty well from other viruses as well, totally as expected. But what we wanted to make sure is that just the virus titer would not explain all the variation that we're seeing, and that's what we see here. So if you look at, for instance, dengue 2, despite seeing like really high dissemination in the wild-type mosquitoes, if we give them high titers, there's still pretty effective inhibition with the two Wolbachias. So that was reassuring to see that even though sort of you give the virus the most opportunity to infect a mosquito, Wolbachia is still decently strong in inhibiting the virus. So far, I've talked about basically the infection and dissemination rates. So that's really kind of population level, sort of zeros and ones. So does a mosquito become infected and disseminated, yes or no? But we can also look at the virus replication in these mosquitoes and basically look at the titers. And so we kind of wanted to see, is there any difference in the replication that we're seeing in wild type versus the Wolbachia mosquitoes? Because previous studies have shown that there's clear reductions in replication where you would expect to see lower titers. In our case, if we look at the mosquitoes that do become infected and have a disseminated infection, we really don't see major differences in the titers that these viruses reach. And so there might be like some slight differences in sort of the median between groups, but overall you can see that the range of these titers is pretty similar for the different serotypes and then between the different combinations. And so that is interesting. So that kind of means that like if the virus is able to basically escape from the midgut, we still see good replication despite presence of the two Wolbachia strains. Another very important factor we wanted to control for is the density of Wolbachia. And typically what previous studies have shown is that the more Wolbachia there is in the mosquito, sort of the more inhibition you can see. And so we kind of wanted to see with our data set, do we see any differences in that density, and could that maybe explain then some of the variation we're seeing? So first here we kind of break it down by the relative Wolbachia density when we test these batches of mosquitoes for the different serotypes. And overall, again, there's maybe some slight differences, but overall the densities were pretty constant, especially for WML. And so that really doesn't seem to explain the variation. And in fact, if anything at all, in WL, we see the highest densities of Wolbachia, and that was the one where we do see substantive viruses that were least inhibited. Another way of sort of looking at the data is by kind of breaking it down by dissemination status. So if you have fully disseminated mosquitoes versus ones that only became infected or ones that did not become infected, do we see any differences there? But even there, it's pretty constant. We don't really see any significant differences, really indicating that we're not really seeing a huge density effect. Now then the question becomes, what does all of this mean kind of from a more epidemiological standpoint? And we were very fortunate to collaborate with Hendrik's group. And so this is really the work from Emily Finch. So this first graph is kind of like sort of summary of the data that I've just shown you. So this is the experimental data where we just showed like a frequency plot of the relative dissemination on the x-axis of the two Wolbachia strains to kind of get a sense of the distributions. And where it stands out that a lot of the viruses had very low or zero relative dissemination with WML, slightly wider range with WALB. So basically what Emily did was using a stochastic transmission model to kind of get a sense for like, what would this mean, this sort of heterogeneity in the relative dissemination mean in terms of like Wolbachia interventions in the field. And so in this model, we kind of play around with two main factors. So this is kind of exploring first, what is the impact of different transmission intensities, so different R0s, and sort of the full range of relative dissemination, like how does that play out in terms of probability that dengue would reemerge in a certain area. And what you can see is that both increasing transmission intensity as well as increasing relative dissemination lead to a sort of faster reemergence of dengue. And so they kind of like, I think intuitively makes sense. But then we wanted to know, how does that sort of play out with the two different Wolbachia types and the 4 serotypes that we tested? So we really wanted to use the experimental data into this model to see how that makes a difference. And so here, you basically see like the 4 different serotypes. And here we model the probability of reemergence within 40 years. The dots basically represent like how many viruses would reemerge. And then the bars are kind of like median time to reemergence with the 95% percentiles. And again, kind of like what we see with the experimental data, what this confirms is that, especially with dengue 3, under both Wolbachias, we see the highest probability of that reemergence. We see some of that as well under higher transmission intensities for dengue 1 under WLB. But overall, there's very few viruses where you sort of would expect to see that reemergence within 40 years when you consider dengue 2 and 4 for either Wolbachia. Then final analysis that we did was basically kind of looking at the selection and diversity of viruses, like how would sort of rollout of Wolbachia impact diversity over time. And that's what we were showing here. So basically in this version of the model, we have 5 strains per serotype that are circulating, so 20 total at the start. And then we're kind of like seeing like how does selection occur and like how many viruses are left and what is then their average relative dissemination. And what you're seeing is that for both Wolbachia interventions, you basically get selection for those viruses with sort of the highest relative dissemination, which makes sense. And so we're really like over this time span ending up with pretty much like 2 viruses, the ones where we see the highest relative dissemination in our experiments that would be selected for. And then just to kind of wrap up with sort of some main takeaways and some discussion points. So I think the first point really to make is that even in wild-type mosquitoes, we see that large variation. So that already drives home the point of like, yes, we need to take genetic diversity into account and not all dengue strains are equal. Second point is that we're then also seeing differences between 2 different Wolbachia strains, with an overall sort of WML being the stronger blocker, WLB the weaker blocker. But again, with definitely differences between specific combinations of Wolbachia and dengue. And that really the key point here is like we see a high level of heterogeneity in inhibition between those different serotypes. And in fact, I could have just said different strains because it's really sort of the variation within that matters here. Then some discussion points that I want to end off with. I think we should really start thinking about genetic diversity in the context of control interventions. So we specifically studied Wolbachia, but I think we can probably take this a little broader, where like, when we roll out new interventions, we should be on the lookout and make sure that like an intervention actually works across the board and not just for, a handful of selected viruses. I would also argue that if a control intervention is not perfect, which I don't think is ever going to happen, there is a chance for resistance to evolve, and this is something we need to monitor as well. So we really need to be on the lookout if we see that incomplete inhibition. We see variation now, but how is that going to change over time? Like, is there going to be more selection? Are we going to see more of these sort of escape strains popping up? And what are sort of the underlying mechanisms? Because that's something we still don't understand very well. And then final point I want to make is that I think it's really important to implement genomic surveillance into the field. So really, the data that I'm presenting is kind of like lab studies linked to modeling, but we really need to make the link from lab to field. And I think genomic surveillance in areas where control interventions are being rolled out is really important to also monitor in the field which strains are we going to see overrepresented in those areas, and what could that tell us about what that means in terms of like how effective our control interventions are. And then I just want to thank my team, especially Afiz, who led all this work, our collaborators, especially Henrik's group, and Emily, who did all the modeling. And thank you. I'm happy to take questions during the discussion.